So after reviewing Owen's RNA page, what I thought was very succinct and well described was the methods section that he put in. It really went into detail what was going on in our lab, and it showed that he took notes of all the lab protocol changes as well as what was really going on in every part of the lab, such as RNA isolation, RNA quantification, what protocol we used, what kit we used. So I thought that was really good. I would recommend though that he adds in some pictures of, the RNA isolation kit we used, the RNA concentration protocol, and a picture for that could just be the nanodrop machine. For qPCR, we could add in a picture of, I'm pretty sure it was the BioRad protocol we did for, I think I might say qPCR. Yeah, so qPCR used a LiveAct protocol, but the cDNA synthesis used the BioRad protocol. So yeah, just include that and then include a picture for each one of them. I just think the only thing that's lacking from this methods section is just making it more, not just a bunch of paragraphs, but also separating it up into, each week, like RNA isolation, RNA quantification, and then cDNA synthesis, and then qPCR, and then putting like an image with each single one of those weeks. That would be, I think, very helpful when looking at this. And then when I read the discussion and the analysis, I thought that he obviously did what was expected about the main goal. He talked about the main goal of this lab. He talked about the results of the nanodrop machine. He talked about how the analysis or the experiments were successful in obtaining, concentration values and actually isolating the RNA. The one thing that I would recommend is talking about how there should be a hypothesis somewhere in this page, and that hypothesis should be talked about in discussion. And the way that it should be talked about is you have to relate it to the results in a way of, do the results support or not support my hypothesis? If you could just add in a quick hypothesis, basically, wondering, does the UV treatment increase or decrease p53 protein, p21 or p53 mRNA, p21 mRNA, Bax mRNA, and BCL2 mRNA, and then explain why you think that for each one of them, and then make a paragraph in the discussion section basically saying, okay, now that we have the results do not support my hypothesis, or they do support my hypothesis, and here's why. That is one part I did see that was missing in the discussion section. Another part that I did see that was missing was the next steps that should be taken after doing this. So we did the RNA isolation, quantification, cDNA synthesis, and the qPCR. But after that, you can still do other things to test the effects of UV treatment on proteins, such as, protein or Western blotting, checking for protein levels of these cells, checking for the protein levels of p53, p21, etc. So just adding in that would be something you have to do for the discussion section. And then for the results section, just making sure that you have a written part of it that starts with to determine if, and that will be followed by what the main goal of this lab was. And what the main goal of this lab was to look at how treating cells with UV would affect the mRNA levels of the 4 proteins of interest that we are looking at. And that was p53, p21, Bax, and BCL2. So if you could just add in that as well with the pictures, and just make sure the formatting is correct with the results, it'll be fine. And yeah, so once you add in the synopsis, and then also just adding in more for the background info of each one of the proteins that we looked at would be extremely helpful as well with images. So other than that, I think it was good, and I really liked especially the paper that he put in the background info, because that's not something that I did, and it was an interesting way to start the paper, and it made it very clear to the reader what was really going on.